knew physics, which was a George's thing as well. And so I think I learned a lot from that, and I hope Padmini and George took something back in return. But it's been a good, so and so I'm really quite proud of knowing Padmini, because she's was one of my I say younger colleagues who's really done very well, and hats off to her, and I wish her the best for the future, and I'm sure she'll take care of this session also in her own meticulous way, bustling, energetic, and but meticulous way. And that's all I have to say.
Oh, so Shahid, thank you very much, and thank you, Ganeem and Ata, for organizing this. So as Shahid said, we had many cups of coffee uh, back in Berkeley when it's such a luxury to think of something like that right now, just getting together in person and having coffee. By way of background, actually, I'm a chemical engineer by training. I'm a phenomenally failed experimentalist. I spent 4 years trying to get things to work experimentally and decided I want nothing to do with them, which is how I ended up becoming a biophysicist. In my background, going from chemical engineering, my PhD is actually in biology, where I studied at a medical school with Ravi Iyengar, learning a lot about signaling. And I recognized, and I say this because I'm hoping a large portion of the trainees and the audience can, back in India, when I went to school in Hyderabad, and 20 years ago now, the notion of biophysics wasn't something that I was aware of, at least in undergraduate education. There was classical engineering and there was classical medicine, and biomedical engineering was just starting to be a thing. And when I was doing my PhD in cell signaling and things like that, the signaling people didn't care about mechanics. Then the mechanics people like Oster, as amazing as he was, didn't really want to touch signaling. So there was this like divide. And in fact, just earlier today, one of these articles that I wrote came out, and I'll pull up the picture at the end as a sort of humorous way of ending this. But, everybody looks at their own little piece together. And it was thanks to Shahid that I actually learned what dendritic spines were. I had not really heard or thought of them before. And it kind of got me into this what has been a very fruitful area of research in my group. So I'm a faculty member at UCSD. I'm in the mechanical and aerospace engineering department. I don't have a mechanical or an aerospace engineering degree, but they gave me the best offer. And, they let me do what I want. And so everybody wins. So today I'm going to try to give you both the research story, but also a little bit of the thinking that has evolved with this and how we can, if you, being an outsider to a field is not necessarily a bad thing, perhaps for the trainees. I think that's sort of my big take home message. So if you're pondering, engineering, cell biology, what is going on? I think sometimes when you come in from the outside, you can have great fun doing things and occasionally make an interesting contribution. So just to set the stage, because this session is thinking about physical design of neuronal signal circuits, I want to get us all oriented on the same page on what systems we are mostly going to be talking about. So if you are even remotely familiar with neuroscience, a lot of what we talk about, the sort of popular science culture of it, much of it focuses on computational neuroscience of networks, of synaptic weight updates and things like that. And then if you talk to neuroscientists, then there are these subsets of neurobiologists and biochemists who are trying to understand the various proteins and their locations and organelles. And even as within each field, there are these little silos, right? So one of the opportunities that actually many conversations with Shahid introduced me to was the possibility of thinking about synapses and spines as biophysical components. And selfishly, at that time, I was actually thinking about problems that if I got a faculty position, I would work on that were not cell motility. Because as far as I was concerned, so many people were working on cell motility, getting oneself established, starting from scratch, being a nobody. This was like, going with the crowd was going to be hard to break through. And so what we're looking at here and what we're going to be talking about for a good chunk of this session is this little guy here shaded in red, which is a synapse where two different units of neurons meet. And neurons, as you know, are large cells with very unique shapes. And one of my own sort of mathematical pet passions is, thinking about geometric problems. And I work on other things in my group in that framework. So what's happening in the synapse, and just to set the stage for you, is it's AM in India, it's 9:30 in India and 8 a.m. in Pakistan a little after. So if you are going to learn anything out of this session, let's say, your presynaptic terminal releasing neurotransmitters, your postsynaptic spine, this is the dendritic spine, which I'll focus on much of my talk, in response to these neurotransmitters, there are receptors, then the channels open, calcium comes in, a series of biochemical events and biophysical events that reorganize the spine. And then this physical action is one of the sort of early steps associated with learning and memory, and is one of the early sort of losses in neurodegeneration or any sort of disease. So you can take these big cognitive things like learning and memory and then map them down to like structural units. So this postsynaptic spine is really thought to be the biochemical computation unit of memory and learning. And what we started thinking about, actually, someone's got to pay the bills for a lot of these projects. And this was actually something that the Air Force Office of Scientific Research had bandied about as an interesting question. So here's the thing. A lot of the, we know electrical signaling, very important. Pumping action of these various pumps and channels is very important, but all of this is very energy intensive. And I want to set the stage because Suhita is going to talk about the energy prescription in a bit. And so when you think about all the biochemistry that's going on in here, depending on your favorite friend in biochemistry, they'll tell you their favorite kinase or phosphatase is important. But what we also learned and had tons of conversations about was the role of actin in the synapse, and the role of membrane rearrangement in the synapse, the role of exocytosis and endocytosis and things like that. And here is something that if you didn't know, this is pretty common anecdote, but there are very good essays if you're considering a career in this field to read an articles by Terry Sanowski and Simon Laughlin on how your brain size by weight is 2%, but the energy consumption is 20%. And this, you will find articles that sort of break down these and tell you that a good portion of the brain's energy goes to maintaining these cells themselves, just to keep them alive. And then 75% is used for signaling, processing electrical signals along the brain circuits. And the bulk of this 75% is actually at these synapses. So what you're looking at in these synapses is actually such rich activity of energy. There have been ideas of thermodynamics of these synapses. So you cannot just keep in mind that as you go through a lot of these processes, and when we build biophysical models, we set black box, we set boundaries. We say system, everything else is surroundings. But just keep remembering that there's so much more to do. I mean, this is what I take away with every time we work on any of these sort of sub problems. There is so much more to do here. And so one sort of idea that you'll see more and more prevalent in the field is the idea of spatial coupling of energy production and energy demand. The firing of an action potential, the synapses, they are extremely energy intensive. And then, when people started showing that the mitochondria are actually parked at the dendrites, at the base of these spines, and their sizes change, they grow to meet the synaptic activity. And loss of energy production through mitochondria can lead to impaired synaptic activity. This sort of set the stage for us to think about some very interesting coupling with signaling, but also some coupling with biophysics. And if you actually look inside a spine, so this object is about 1 micron cubed in volume. It's extremely densely packed. It has this feature known as the postsynaptic density, which is a very rich agglomerate of proteins, and there is some preliminary evidence. There are a few papers that talk about even the liquid-liquid phase separation in these regions that seems to be critical for localization of signaling function. You have actin, and about 19% of these spines, these mature spines, have also an ER in them, as a specialized ER known as the spine apparatus. So if you just think of something as simple, it's not, but if you think of something as early as calcium, its regulation is so robust, and so many modalities of robustness. And honestly, if you look at it from the outside, and if you're not a biologist, you might say, well, anything that can happen will happen. That's how messy and complicated the system is. And this is sort of partly true, but not anything that can happen does happen. In fact, you have very nice studies showing the pathways associated with memory and learning, the notion of a long-term potentiation or long-term depression, where LTP reinforces pathways and LTD weakens the pathways. And if you go from these ideas of reinforcing, updating synaptic weight to weakening pathways, to thinking about what are the biophysical changes, then you have that LTP is actually associated with the change in the spine volume. This was the focus of the paper that Shahid and I worked on. There's a change in the spine volume with LTP. So within about 5 minutes, this object, which is 1 micron cubed, changes its volume to 200%, and then it decays back. It doesn't reset fully to 0, but spine rescaling occurs during sleep. So and this is when you kind of go from short-term memory to long-term memory. So there is the whole span of circadian behavior here. And there are projects that I won't have time to talk about today on how we are trying to couple this with Ralph Greenspan's group on the circadian behavior. What has that 24 and a quarter cycle got to do with these sort of short-term signaling events and things like that? So today, in this session, I will talk about the work that my group's been doing, and then Suhita will talk about this very nice series of papers she's been writing on the energy information prescription. So I hope with some of this background, you can start to see why that is important. And Khaled, who will join us from Qatar, will talk about calcium tunneling and how that can play a role in localizing in calcium signaling modality. And then Ganeem will talk about what all of this means for Alzheimer's disease. So you can see the whole spread from sort of basic to Alzheimer's disease. And I must admit that it is close to 8:30 p.m. in California, and I'm not a night owl. So at some point I will excuse myself. And so if you have any questions or you want to discuss this further, please email me. It is not for lack of respect. It is, you don't want to talk to me beyond a certain hour. It's completely useless. With all of that said, here's my contact information. And I'm going to actually get to the scientific aspects of what my group's been working on. And this is kind of the sort of problem I'm trying to tackle piece by piece. There is a version of this Shahid in our paper. This is an updated version. There's a version of this that we've been tackling. So basically, if you think about something like long-term potentiation, which occurs over a 1-hour time period, you back up, you see there is sustained spine enlargement. There's the transient spine enlargement that happens over the 5-minute time period. You cannot change these sizes and shapes without working with actin rearrangement. So you have all of this. Now you're looking at these timescales here. But other things are happening when we talk about reinforcing pathways or weakening pathways. What's happening is you have receptors, AMPA receptors, which is a receptor for a neurotransmitter glutamate, is, you have more receptors insertion and clustering at the postsynaptic density. So in parallel to the cytoskeletal reorganization, which we are working on, you also have the receptors that are, either diffusing in the plane of the membrane or being exocytosed, being trafficked to these regions. And this is, there's a lot of beautiful experiments by Daniel Choquet's group in France that, lend themselves to beautiful modeling. Much of what I'm going to talk to you, so we have ongoing works on here. So in about the next couple of months, you can expect preprints on bioRxiv, but that's not what I'm going to talk to you about today. I'm going to talk to you about our logic of how we've been building these pieces of the puzzle. Okay. So when I talk to you about a neurotransmitter being released, the first thing that comes into the neuron, into the spine, is calcium. And that's your ion of all important significance. And calcium is coming in through these two receptors, mGluR and NMDA receptors. But in parallel, you also have cyclic AMP activity. Cyclic AMP and calcium then regulate CAMK2, which is a very important kinase in these spines. And not only is it just your good old kinase, it's a beautiful kinase because it's a dodecamer. It autophosphorylates, it does its own thing, and it's an actin bundling protein. Okay, so it actually plays a big role in how the actin's rearranged. And then on this side, you have pKa, and pKa protein kinase A downstream of cyclic AMP is very important for, sending signals to the soma saying, hey, I need proteins made or I need material delivered. So we're going to talk about this a little bit. I'm going to take you down step by step. So we got interested in this calcium influx. Actually, not so much because we were interested in calcium, because we were interested in a specific question that had more to do with spine shape. And I'll show you how we sort of parse out our own thinking when things get messy. One thing that we'd found in the literature was that these dendritic spines have very well-defined sizes and shapes. And they have been classified, and there's tons of papers written on this, in terms of, the mushroom-shaped spines are, they are right protected memory spines, and the thin spines with spherical heads are reed spines, they are learning spines, and things like that. So we actually wanted to address this question. What does the size and the shape of the spine mean for calcium influx? Now, you can't just take this question and, write yourself a model. If you're talking about calcium, given the rich history there, you have to consider the key features that play a role in calcium dynamics, the pumps, the channels, and the buffers. And now you also have to worry about diffusion and the location of these spine apparatus that I told you. So my student, Miriam Bell, we actually did two things. One is we started thinking about the simplest thing that we can do. So my favorite kind of thing to do is to write PDE. So we just wrote the simplest PDE that one could think about, in which you have diffusion of calcium in the volume. There is some reaction rate that accounts for, there's some time scale that accounts for all the details. And you can keep track of the evolution of calcium over time. Now, here's the trick, though. If you have a mathematical bent of mind, I give you this equation. And if I give you Dirichlet or Neumann boundary conditions, you'll write the analytical solution subject to some coordinates and separation of variables, perhaps, and then you'll be done. And you can do that for geometries like these. The kicker is that once you put your boundary conditions, you have now time-dependent flux boundary conditions. And these fluxes are what become important, you see, because it's not so much that calcium is diffusing in the volume, but it is that it is coming in through these boundary conditions mathematically, which are your plasma membrane boundary conditions or your ER boundary conditions, okay? So once you put those in, and then we actually simplified this problem quite a bit. We said, okay, we're going to just write fluxes that are time dependent. And we have two geometries, the spherical spine head to represent the thin spines, and these ellipsoidal spine heads that are good enough approximations of mushroom-shaped spines. So if you take these equations, you can write out actually using separation of variables, you'll get spherical harmonics in this case. Here you'll get ellipsoidal harmonics, and then you can actually play with these parameters of these sizes in there. What was the point? The point was very simple. The point was, depending on your parameters of the eccentricity of the ellipsoid and the relative eccentricity of the outer ellipsoid versus the inner ellipsoid, you could get hot spots of this component C, which we is a surrogate for calcium. You could basically get hot spots here. And if, as you change the size, nothing, the chemistry is not changing. That was never the point. Your, peak moves over here. If you're familiar with harmonics, you would say, well, that's not surprising. You're just redistributing the wavelengths or moving things around. And that is true. But from a consideration of, if you then have downstream events, which, in that big flow chart I showed you, you then have CAMK2, pKa, what have you, these little timescales could become very important on where your reactions might occur. So this sort of gave us the first insight into, maybe shape is important. So then we did the next thing, which is we parametrized this model for more realistic calcium dynamics. Rather than just putting, fluxes out of nowhere, we actually considered the NMDA receptor, the voltage sensitive calcium channel, all of the nonlinearities that had been studied and proposed for many years. So now on the plasma membrane, you have all of the plasma membrane fluxes. On the spine apparatus, which is the specialized DR, you have these relative fluxes, and you can localize to the particular PSD, and you supply a membrane voltage, and this is your stimulus. You have an EPSP and a back propagating action potential, and then you ask what happens. So first, let me get into this. The first thing you see is that the spine head volume. So we were primarily trying to see. So I'll let you in on a secret now because I know the answer. Back then when we were working on this, we were actually trying to understand if geometry even mattered, and if so, how. And so we had two approaches we could take. One was we could work with people who are doing electron microscopy and extract those realistic geometries and reconstruct them and use them for simulations. At that time, it seemed a very expensive, daunting adventure when we didn't even know if this was going to pan out or if it was relevant. We had no idea of any of these physics, you see, because all the models, to our knowledge, except one that was published by Tom Bartol, had sort of well-mixed ideas. So we had no idea if size or shape even mattered when we put in the realistic details. So what Miriam did very bravely, first year students can be very brave because they don't sometimes know better. Very bravely varied the volume of the cytosol, spine cytosol. And we found that the peak calcium that you're looking at essentially decreases with the spine volume. And that decrease can depend on the shape. Now, again, idealized shape simply because we wanted to make sure our solutions were relevant. But more importantly, what she found was that the distribution of these membrane fluxes. So now I should say there is one thing you can do with these kinds of simulations that is a bit hard to do analytically. You can localize components to certain regions of the spine head. And you can say, my NMDA receptor is only at the PSD. So as you can see here, the NMDA influx is only at the PSD. Let me get my annotations. Whereas the voltage sensitive calcium channel is everywhere. So you don't see these localizations. And so one thing that we kept coming back to is that these localizations were actually very important for spine calcium. All right, so this kind of, I should say, I'm quite happy if somebody wants to interrupt and ask questions. We don't have to go through everything. I'd rather we go through in detail. So if the audience wants to interrupt, I don't mind at all.
Yeah, I have a question. This is Aman, just a quick question. Padmini from George Mason University. Sorry to interrupt, but it's 11:45, so at 35. So if I misunderstood you, please correct me. Did you say the voltage channels are spread out or it's concentrated in that region?
So the voltage sensitive calcium channels are actually distributed everywhere on the spine head.
Okay.
The NMDA receptors are focused only on the PSD.
Thank you.
Yeah, sure. So, okay, so now we kind of, remember I told you something very interesting about mitochondria. So then we kind of got into this. So we like looking at microscopic pictures, even though we don't like doing experiments as a group. So we actually encountered this notion of ER mitochondria contacts. So this is something that is an emerging field in, interorganelle contacts. And I should mention that the other half of my group that doesn't care about signaling cares a lot about membrane mechanics. So we do think about the physics of how these organelle contact sites come about. So here's what you're looking at. The green is the ER. This is from Gia-Wolz's group. And the purple is the mitochondria. And you can see these like little ring-like structures and these making a contact. So we got quite intrigued by this and what it meant for calcium dynamics. So these ER mitocontacts obviously have a lot of proteins assembled there. But if you try to draw a physical picture of it, what it is simply this red region here is a conduit. It's a direct tunnel to go from one organelle to another. Okay, so it's basically reducing all these flux barriers. So we actually started thinking about the coupling between energy and calcium. So this paper, it's in final round of revision, hopefully. And there's a preprint on the bioarchive, but essentially we wanted to look at if you had ER mitocontacts, what do you gain in terms of the ATP? And is it obvious? Now, if you look at a schematic like this, you can put all your mitochondrial fluxes and you can say, well, yeah, I mean, if you have a ER mitocontact, I don't even understand why you need to deal with this problem because more calcium goes directly between the ER and the mitochondria, you get an immediate like increased ATP response. So what's the question you're seeking to answer? And I'll show you how some of this, our own thinking on this evolved. From a technical standpoint, it is not trivial to build spatial models of signaling, let alone with various organelles. So I will give Alan a lot of credit for actually building these geometries, getting the numerics to converge and things like that, because it's not trivial at all to do this. So we have now what we are calling MERC, which is the ER mitochondria ER contact site. And we have in this little conduit, you can start asking these questions about what happens with and without contacts. So first, you look at calcium and IP3, you notice that it's actually, nothing to write home about, right? Your calcium in the cytosol, your IP3 in the cytosol, you can look at all these gradients, they're pretty, but if you look at these comparisons, there is really nothing to write home about. But what happens, and this is very interesting, is that the mitochondrial dynamics are different. So what I showed you on the previous slide is the cytosolic calcium and IP3, but the mitochondrial dynamics actually are quite different. The mitochondrial calcium, because you put these little tunnels having a direct flux of mitochondrial calcium, the mitochondrial calcium is higher. And what this does for you is it increases both the peak and the rate of ATP production in the mitochondria. Okay, so this is just tuning the rate to be faster by putting more calcium in. The cytosolic ATP, which now needs to be effluxed from the mitochondria to the cytosol. So bear in mind, you're making the ATP in the mitochondria, and then it needs to be effluxed through a series of proteins into the cytosol. There is a little bit of a delay, but if you have these contacts, you then end up getting more cytosolic ATP. Okay, phew, because, at least something happened, right? But interestingly, it's not enough. This is where things start getting interesting, because one of the things that we noticed in the literature was this whole range of mitochondrial sizes in the post-synaptic dendritic region. And so your mitochondrial calcium seemed to respond proportionally to the mitochondrial size. So here we are increasing the size. Oops. We are increasing the size as we are going in this direction. And so, of course, your calcium in the MERC increases, and your mitochondrial ATP also increases. And there is some nonlinearity here, with the second bump, and we're doing some sort of investigation on these sort of dynamical systems. But here's the thing. If you take this very small mitochondria, your cytosolic ATP is actually not increasing. And this really comes down to making the ATP is not enough. You got to have enough surface fluxes to get it out into the cytosol. And as you increase the size of the mitochondria, the net surface area available for these, you can get an increase in the cytosolic ATP. So now we're starting to get into some interesting ideas of what is a good mitochondrial size ratio that you want.
Hi, Sriram here. I have a question. I didn't see a way of raising my hand, so I'll just ask it.
Sure.
In the PDE that you start with, you have a time scale tau and you have a diffusion. Therefore, there's an intrinsic length scale, square root of diffusivity times tau. And presumably it's important where these sizes lie with respect to that length scale. So can you give some idea of how much that length is and where these lengths lie?
So the diffusion of all of these components is very fast. In the original PDE, I should say this, in the original PDE that I showed you where I only had that one component, we subsequently actually added a lot of these details of these various proteins of, these. So I couldn't extract for you a single time scale here. But the diffusion of calcium is roughly, 300 microns squared per second or something like that. So it's a very rapidly diffusing. And so is ATP. It's of comparable. It's small molecules comparable, unless you think about electrostatics and crowded diffusion, which there's no evidence that they're trapped in these cases.
Yeah, but that time scale tau in your initial PDE is gone from the rest of the analysis, or what? You had a
no, the time scale tau is now accounting for all of these sort of kinetic species that are in here.
So, but then with that time scale and a typical diffusivity, you will get a length. So that length is, what I want to know is how that length compares to the kinds of size
of the mitochondria. Yeah. Here, I'll tell you right now. So your diffusivity is about 300 microns squared per second, let's say, and your time scale of calcium, we can sort of extract these time scales from any of these. So you have, say, 10, 15 seconds. So what do we have? We have square root of 20, right? So 4 odd microns, roughly. Okay. So yeah, I think sorry, go ahead.
No, go ahead.
No, I think what the issue here, what becomes rate limiting in this case, Sriram, is that the surface area. So if you know those cylinders, the surface area and then the flux rate, right? They start competing. So it's not that you're not making the ATP. In fact, here you can make all the ATP you want. You can't push it out fast enough because you don't have enough surface area into the cytosol.
I see. Okay. Thanks.
Yeah. And in fact, I should say that we've actually been we've been calculating the surface areas of mitochondria in realistic geometries, but we'll come to that later. So I think this was something we kind of ran into. But there's one other geometric feature in this here, which is a very well-established variable in biology, it turns out, that we weren't aware of, which is the area fraction. So if you look at this little gray torus here, the area fraction that is shared between the mitochondria and the ER, that seems to vary quite a bit in cells. So that would be essentially changing these relative, area fractions. And so that was one other thing that we changed. And, again, it kind of comes back to this balance of the following. So if you take so there's a lot of data here, but I could tell you this in a couple of sentences. If you take a large mitochondria, you can make the large ATP, and you can even efflux it. But if you're in a better position if you have a larger area fraction. So these area fractions, I should say, are actually informed by experimental observations. There are about 30% of area fraction. So within a given mitochondrial length, the area fraction doesn't matter, but you need to have a larger mitochondria to have more cytosolic ATP. So this kind of comes back to the various boundary conditions and the fluxes that compete with each other. And so that this was sort of where we leave the story. It's, you know, it's very new. It's something we're thinking about in terms of what this means for the geometry, which is you know, a lot of these metabolic, I'll tell you why I care about geometry is because a lot of these metabolic parameters, you can find them in the literature. You will be able to find cellular consequence. We are more interested in what these geometric parameters kind of translate to in terms of system effects. So one thing that you can ask almost immediately is, you said these sizes are so small and you wrote a bunch of differential equations. Like, how is that even valid? Like, can you define a concentration in these cases? So this is a very valid point. In the defense of differential equations, I would argue that if you don't see interesting things in sort of, you know, deterministic limits, then it's, I mean, I like deterministic problems, and then, you know, we can think about them in their stochastic framework. So this actually is something that this is a work that a high school senior has been working on, and we are putting together a manuscript. So I just want to highlight, you know, when I was a high school senior back in India, I was watching Bollywood movies, but you know, there are high school seniors who are writing research papers here. And so Mason used MCell, which is something that Terry Sinowski and Tom Bartol have pioneered, to take these same problems of spine sizes and shapes and actually run stochastic simulations. So this is very, very new work. These results are like new as of like yesterday, some of these. We're very excited about it. So here again, what we did was we actually took these fluxes that I talked to you about. All of these are now in a sort of more realistic geometries than what I showed you before, where you think about these thin spines, these mushroom spines. In fact, if you look here, you can see these little depressions that, you know, represent the sort of mushroom head, and actually asked, how do these sizes and shapes, if at all, correlate with the synaptic learning rate? So there's this classic model by Showal et al. On what represents the learning rate and how you can update the synaptic weight. Now, those models do not account for geometry, so we kind of wanted to play with that a little bit. So what Mason did was, in all these combinations of geometries, organize them in a way that we can first think about, think about sizes. And so he started with these filopodia shaped geometries. The reason, you know, we did these filopodia shapes is this is when the spines are trying to like explore their environment and figure out what's going on. But they were the simplest geometries that you could sort of think about learning how to set up these simulations. So I'll walk you through this because these slides have a lot of information in them that I'll walk you through and distill, and then we'll see if we can make sense of them. So he takes spines of 3 volumes. All of these volumes are informed by literature measurements of what spines should be like. And for each shape, we run 50 seeds, so 50 simulations. And these are 2 individual simulations here, 2 individual random seeds. And you can see that at 15 milliseconds, you have 263 calcium ions, and then at in the other simulation, you have 437 calcium ions. If you increase the size, you see this numbers change, but they're quite variable because we're running stochastic simulation. At 30 milliseconds, there's practically no calcium left, and the numbers are quite small in these systems. What was interesting to us, and this is something that, you know, in a well-mixed, excuse me, not in a well-mixed, in a deterministic sort of continuum representation, you wouldn't see these sorts of necessarily these patterns of, but what we kept seeing in these filopodial simulations was these sort of plug flow-like behavior. The entire calcium domain is like a plug that just goes together. It kind of moves together in here. And if you look at the mean of these calcium ions and then the variance, what was very clear to us was there was a size dependence on both the peak calcium and the decay time. So you can see that here, the peak calcium is increasing. In this case, we looked at volume. We also looked at volume to surface area, and the volume to surface area had a better correlation. And the decay time constant, which is how this calcium is decaying, it also has, it decreases with the volume to surface area. And correspondingly, as you increase the size of the filopodial spines, your synaptic weight is updated and increased. So this is it's a metric of learning, if you will. So if you take now, you transition to a thin spine and you repeat the same idea, you now have different volumes because your geometries are different, obviously. And you can see that, at early time, the whole thing is more or less in the head, and then it starts to dissipate. And regardless of the spine shape, you can start to see these same behaviors. Whether you have the peak calcium, which starts to be linear with the spine volume to surface ratio, or the decay time constant starts to decrease, increase in synaptic weight for the larger spines. And then finally, we kind of did this for the mushroom-shaped spines. Now, in the mushroom-shaped spines, these geometries actually we calculated using a mechanical force balance. And so in the mushroom-shaped spines, again, you can see much larger calcium. You can see the peaks, you can see the decay time. So one thing that was very, very surprising to us is across these stochastic simulations, across shapes, there seem to be these repeated behaviors of the peak calcium and the decay time constants of how they varied with this volume to surface ratio. And we even added the spine apparatus as a way of excluding the volume. And you can see here the spine apparatus is in there. Because of the 3D projection, we still haven't been able to figure out how best to get rid of the calcium in there. But the same relationships hold. So what is the sort of point? What we're doing right now for this study is we have these realistic geometries of spines we've picked up from the literature. And we are trying to see if these relationships that we're talking about with respect to these volume to surface areas hold even for realistic geometries, or is it something that just we stumbled upon because of the idealizations of our geometry? So the story is not complete yet, but we think we may be able to predict some sort of a deterministic behavior across spine geometric properties, in this case, volume to surface area, as a function of, for their calcium behavior rather than focusing heavily on just the shape. So stay tuned. So how are we doing on time, Shahid? I could talk about the cyclic AMP work, or I could take questions. I'm okay either way.
Well, Ghanem, Ghanem or Atta, you are you tell us.
I think we should give the audience, I'm sure there are some questions, so maybe we should give them some time. So if you tell me, could you wrap it up maybe?
Yeah, okay, so let me tell you this. So let me tell you then that these were the papers that Shahid was referring to, where we actually coupled calcium to cyclic AMP. And actually we found some very interesting frequency filtering behaviors. So what I'll do is give me a minute, and I promised you I would show you something fun, and I will do that right now, and then I will open up for questions. So where's my fun thing that I wanted to show you? I had it on my desktop because I took a screenshot of it. And I will leave you with a fun movie and then a fun picture. And we can talk more. Well, you can ask questions and I can stop talking. So you can see my screen again. So the point that I'm trying to get at is a lot of what we've been trying to do is get at these sort of mysteries of size and shape. So I told you we can work with realistic geometries. And so this is a full reconstruction of an entire dendritic branch. The data are originally from a paper that Pietro Di Camilli published with his group members. And that was basically FibSum data, okay? That was just microscopy data. So Chris Lee, who's my postdoc, and Justin Laughlin, who's my student, they took that data and the reconstruction, put all of it together, meshed it, made it compatible for finite element simulations. And what you're going to see in this movie is a simulation that we have generated of calcium influx on these spines that is capturing all of that biochemistry I kind of went on about, but this time in this entire dendritic segment. And this is why we can now work with these realistic geometries. And so you can see when there's an action potential, the spines light up, more calcium comes in, and over time it slowly decays. It's just great fun to watch. I don't have any big scientific insight other than we can actually run these simulations. And in terms of fun things I promised I would share, and this is sort of a thing to think about, I think, for trainees in the audience. You know, when you go into these interdisciplinary fields, how do you deal with challenges? So we were asked to write an opinion on spatial modeling of signaling, and I kind of wanted to put an elephant in there, so I did. So, you know, here's the deal. So much of, when you think about many of these problems in cell biology and biophysics, it's very easy to get siloed. It's very easy to say, you know, I care about the cytoskeleton, I care about the membrane, I care about a channel, I care about whatever it is that I care about. And sometimes if you end up having this view, right? I mean, from the subcontinent, most of us know the story of the blind man looking at the elephant. And this is, in fact, what I was trying to convey here. If you get too siloed into these pieces, you know, you can sort of perhaps miss the big picture. So one of the sort of challenges in dealing with any of these complexities is forces matter, geometry matter, signaling matters, they all matter. And so we do have to keep pushing creatively as a community to sort of really not do this, but actually think of the big picture. So this is just it was an opinion piece, so, you know, I could stick an elephant in there. So I will stop here and be happy to take any questions that you guys have on this. Yeah.
Okay, so questions? Thanks for a very interesting talk. I'm sure there are a few questions. So, Sriram, you go ahead.
Yeah, so at some point in the discussion, you said that you've calculated the shapes of the
spines.
The spines using force balance. So this was membrane mechanics, or membrane plus cytoskeleton, and what was balanced against what? Because
so this is yes.
In
so
the
this I can.
You gave, you had fixed geometry, but then you said this.
Yeah. So actually, where I started this whole problem was with Shahid. Actually, was I wanted to couple the actin membrane mechanics and watch the thing deform. That turned out to be a much bigger problem than, you know, I could take on at that time. So we wrote some simplified equations. So I can show you actually what we did I have it pulled up right here. So what we did was balanced membrane mechanics, as you rightly got at. But we used these heterogeneous forces based on what we knew from the literature about actin. Okay, so this essentially gets us to thinking about, for example, in the mushroom-shaped spines, we know that there's a deviatory curvature along the neck, and then there are actin forces that are balancing on the head of the membrane. So we basically did what I would call an inverse problem. What would it take to get these shapes that are known? And so we have sizes and shapes. There are textbooks written about this, so we don't have to make any assumptions here. And from the model development standpoint, it's a lipid bilayer, your classic Helfrich that you know, and then we use force balances locally to extract these shapes on there. Something that is in the works is using Cytosm, the Francois Nerlec's software, to obtain now with these shapes. So what we get out of here, you can do all your energy minimalization, force balance, the usual thing. What we get out of here are these shapes, and what are the relationships between tension, ill-defined, right? Cortical tension, some form of tension, and then the area of the applied force. What must be the, you can do a back of the envelope calculation of number of actin barbed ends and things like that. So all of that we did. So now what we're doing is taking these shapes as input and asking what would be an actin network that you could sort of seed into these shapes. So this we are doing with Cytosm. And then the next step is to deform the shape with response to actin loading. So this is the sort of thing we wanted to do, but then everything is coupled to calcium, you see, because calcium, CAMK2, actin. So then we kind of got into that little side territory. Yeah, thank you.
So, Padmini, I have a question.
Yes.
So this, so no, I mean, this is great. At some point, I guess, I mean, you may already have gotten into it. There's this issue of membrane vesicles being inserted or being taken out, and that's the kind of important window to maybe start looking into. That now you seem to have the tools to sort of do that.
Yes, so I can show you some work we've done on endocytosis. It was not necessarily with respect to synapses, obviously, but the sort of principles hold for a large part. And so this is something we published now many years ago. On what happens for clathrin-mediated endocytosis. And so some of this, we've been talking to Erik Jorgensen for his ultrafast endocytosis measurements and synapses. And Shigeki Watanabe, who was Eric's postdoc and is now his own group, to think about these ideas. So we do have these the various aspects, right? There's the signaling that drives the actin, that drives the shape change, and then you have the membrane trafficking. So there are all these little pieces. It takes a lot of creative coding, really. I think the physics almost like many of us know what needs to happen. I think the implementation to realize this can be a coding nightmare. It requires some really brave postdocs to sit there and implement.
Yeah, so it's getting the simulators,
Yeah, so I think for a lot of these sort of physics-based problems, so here we only cared about, this is the Helfrich model, we only cared about endocytosis, we only cared about the shape and tension. They had nothing to do with synapses or anything else, right? So, and then subsequently we wrote something about endocytosis and actin. This was published last year, where we actually simulated the actin filament. So I kind of feel like, sometimes I live two lives, one where I'm agonizing about calcium and calcium buffers, and the other days I'm agonizing about what are the force balances on the membrane and how do things change. But I think there's the secret identity is I kind of want to put them all together. I need really, really brave people who will sit there and deal with, details of the biochemistry and with the details of the mechanics, because the mechanics is not trivial either. Getting people to be thinking about that is not trivial either. So,
More questions? Actually, so I have one kind of numerical question and then one biology question about the microdomains between the ER and mitochondria.
Yeah.
So the numerical question is the easy one. do you develop your own algorithm for the finite element method, or you guys use some program which is already out there?
So for a lot of, if you just want to solve reaction diffusion equations in geometries that are nicely measurable, then we just use either Phoenix or COMSOL. We don't try to write our own algorithms because then we'd have to prove convergence for all of these algorithms. For the last movie that I showed you of those realistic geometries, we actually used Phoenix for the simulation, but we wrote our own meshing software to get those geometries to be compatible with finite elements. Because if you take any of the FibSim reconstructions, they are not, you don't get regular meshes, no simulation will converge. Your Laplacian operator will break on them. So we wrote a meshing software. It's called Gamer 2.
So COMSOL, I know, just I have never used it, but I at least know the name. Does it allow you to? So I know the simple examples. If you know you define your boundary conditions and you have your PDEs, and you basically run your the solver there. But if you have stochastic processes, which where you basically they change with time, right? So you don't know when something is happening.
You can't use COMSOL for stochastic. COMSOL is fundamentally just a PDE finite, like it deals really well with the Laplacian operator and nothing else really. I mean,
Okay.